Yes.
Hi, I'm Hui, a PhD student in the Bruce Lafroy Center. So I'm working on a neurodegenerative disorder called Friedreich Ataxia, short form FA. It's due to genetic alteration in the genes where patient has got expanded this triplet GAA repeat, and then it causes them to have a lot of symptomatic changes in their entire body, where they've got gait issues, scoliosis, which is a backbone issue, speech issues. So it's basically, it affects everywhere of the body. So this is a Bruce Lafroy Center. Yeah, it's sort of all the way there. So Bruce Lafroy Center works on a lot of neuro genetic diseases. So there's people working on autism, there's people working on Parkinson's, and I'm working on FA. So my research is trying to develop cell and gene therapy to treat FA. So I work on a FA mouse model, and so what I do is I do bone marrow transplant into the mouse to see if I can correct the.
So do the mouse have FA or you?
Yeah, the mouse have FA. So we got this mice from the Jack's laboratory. So the Jack's laboratory is this really big mouse laboratory in the US, which has got a lot of different kind of mouse models that you can just buy from them and stuff. So we got this from them. No, it's a FA. Yeah, it's not a normal mouse. So it's a mouse which someone has managed to created it to become FA, to have FA. So they deleted the mouse FA gene in it, and then they inserted the human FA gene, but it's an expanded one. So it's sort of what do you call it? It's a chimeric mice. So it's sort of, they're trying to make it looks like it's a human disease in a mouse. So yeah, something like that. So then so by using this mouse model, which is the best available one at the moment, we're trying to see if by doing bone marrow transplant we can increase the frataxin level, because in FA patients the main problem is that patient has got reduced frataxin protein level. So what we want to do is to see if bone marrow transplant can increase the protein level, and to see if it corrects the behavior. So we then test our mice on different behavior analysis, like a rotating rod. So we put the mice on the rod and see how long they take to fall and stuff. So then we measure the latency for them to fall, and then we test them on like a open field. So we just see how long they roam in it. We just let them roam in there for like 10 minutes, or some people can do it for an hour. It's really up to you. But then according to a lot of publications, a lot of people have been doing 10 minutes, so that was what we did. So then there's like a computer analysis system which then analyze everything for you. It records and tracks when the mouse is in there, and so then you can look at the speed they are moving, the total distance they move in the total period of the 10 minutes, and also the latency for them to go outside or inside. So it's a really big box, but then we can sort of draw boxes. So it's a on the software you can draw boxes, so it's not actual boxes in the boxes. So then you can see how long they take to go into the outer box or the inner box, something like that.
Okay.
So that's basically what we do. And then what I've been doing today, so after we test our mice on behavioral tests, and we then cull them after that.
Yep.
And then we collect tissues and stuff, and because mice FA patients are affected everywhere, as I've said earlier, then we collected various tissues. We collect the brain because they're affected in the cerebellum. We collect the liver, we collect the heart, because you know how they get cardiomyopathy and they die mostly because of that. We collect the spinal cord and the dorsal root ganglia, because they're affected in the dorsal root ganglia. It's one of the very main neuronal cells which are affected in FA patients. And so we collect all of the tissues, and then what I'm doing today is called Western blotting. So it's a technique where you can then quantify protein, look at protein. And so what I'm doing today is I'm running spinal cord samples from my transplanted mice to look at frataxin protein, to see if there's an increase, or if it changes the frataxin protein following the transplant. what basically I do. And yeah, and apart from looking at protein, you can also look at RNA level, so frataxin RNA level, which is the level before the protein is being made. So from your DNA you get RNA, and then from your RNA you get the protein. So yeah, you can.
So how far from in the lab a discovery to on the bench?
On the bench, yeah, I get this question a lot. It's probably going to be another 10 years, maybe 5, 10
But we're getting
years.
there, we're getting there?
Yeah, we will get there eventually.
But money would make it a lot quicker?
Of course, yes.
So people need to give?
Yes, because we need to first do even more mouse work to ensure that what we are seeing is true. Then we need to go into the preclinical trials, where then we start testing on patients. So it's clinical trials, and then there's different stages of the clinical trials which it then needs to go through, and then finally it then is available to the patients, to the wider population of all the FA patients in the world. So, and in each stage you need millions and millions and millions of dollars. So it's definitely more funding would be better.
Yeah.
Yeah, and it's really expensive to run.
To run a facility like this here, yeah, this
yes,
is...
and even to keep a mice,
Yep.
I think it's $6 per day.
$6 per day to have a mouse?
Yes. And so I've kept my mice for a year and a half before I cull them. So I test them at different ages after the transplant. So calculate that's how,
That's
yeah.
a lot
of money. Just
because it's not just because you haven't just got one mouse.
Yes, I don't only just have got one mouse, so yeah, it's a lot of money. So just by keeping those mice alive, it's really, really expensive. So for us, we need to try to save money at the same time, so keeping the colony as small as possible, but also to ensure that there's enough so that it's statistically significant.
Yep.
So yeah, it's really hard with we need more money, definitely. It will be so much easier.
Well, I'm as you know, I'm rowing the Tasman this year, and I'm hoping to raise at least $250,000 doing the row.
Yeah.
If I could make $500,000 doing the road, that'd be awesome.
Yeah, yeah,
of course. And that would go a long way in here.
Yeah, definitely. Yeah.
Well, thank you very much for having a chat with us today.
Thank you.
Been a pleasure.
Yes.
Keep up the good work.